All right, thank you, Lindsay. As I wait for my esteemed panelists to join me, disclaimer. Okay, so I'll introduce myself. I'm Fraser Bocell. I'm a senior clinical outcome assessment scientist with the Critical Path Institute. I'm joined today by my colleagues Kevin Weinfurt, Kim Cox, and Andrew Trigg. Our agenda, my introduction is not the introduction. Kevin is going to be giving an introduction to the session. Then we'll have a panel discussion where we will walk through nuance, which will be explained later, and then we'll leave some time at the end for questions from the audience. So, Kevin, if you wouldn't mind, come join me up here.
Okay, good afternoon, everyone. Oh, sorry, I'm very enthusiastic. I'm going to give a sort of a high-speed, high-level, historical, borderline tedious funnel into our topic today. So going way back to around the 80s, there was interest in trying to understand how patients were feeling and functioning, something that we called meaningful aspects of health, thanks to a 2015 ISPOR task force. And the main approach for getting at that back then were self-report measures, which we now call patient-reported outcome measures. And the reason we're trying to do that is we'd like to understand how some intervention has an effect on the person's day-to-day life. How will they work with, sorry, it's very hard to see that screen when this is over here. All right, I'm going to stand up this way. We'd like to know whether some product has a favorable effect on how patients feel or function, which is how we describe clinical benefit, right? So we've got patient-reported outcome measures is the main thing folks were focusing on back in the 80s, and they would generate a score, and you know, that score was often on some metric that was unfamiliar to people, and so it was hard to figure out what to do with that. And so in the 1990s and early 2000s, a lot of different approaches arose, both qualitative and quantitative, to try to help translate PRO scores into something more relatable to patients' daily lives, because that's what we ultimately are trying to do, is figure out, well, how would this patient be on this treatment versus a comparator, right? So there are a lot of these different methods to try to help with that translation. Over time, the field narrowed its focus for various reasons on one main method applied to one type of PRO-based endpoint. And that method is the anchor-based approach using patient global impression of change or patient global impression of severity as the anchors, applied to a change from baseline score, which is a continuous score. Right? And so you effectively get a responder analysis when you do that. Right? So this was the method that the field sort of moved toward and followed for many years. Also over that time, though, we've had a broadening of the approaches for trying to get at how patients are feeling or functioning. We've had additional types of clinical outcome assessments, right? And we've had additional types of endpoints. I've just listed 3 here, but there are many, many more types of endpoints that could be constructed with COA scores, right? Now, the dominant methods for translation that people were tending to use didn't always work that well for this broader range of COAs, broader range of the concepts that they measured, and the broader range of endpoints. Sometimes that method didn't match the endpoint particularly well. For example, if you've got an ordinal score, change from baseline on an ordinal score is not particularly well defined or interpretable, right? Because you can't subtract ordinal scores to each other very well. There were violations of assumptions that made those translation methods work, and the results you get from them sometimes just weren't very compelling, even when all the numbers looked right. It didn't give you a sense that we really understood anything better about how patients might be on this treatment versus another. And so the PFDD Draft Guidance 4 encourages consideration of a wider range of methods, so sort of going back to opening up a variety of approaches to support endpoint interpretation. Right? Now, when you do that, it's good to try to get a handle on all the different ways of doing this. And one way that we could do that is to sort of sort these into two categories based on how they do the translation. What's their strategy here? So one strategy is to take COA scores and to translate those into something I understand better, right? And that's called meaningful score regions. The region word is a little confusing and misleading there. There's a broader range of things than just regions, right? But the idea is, okay, I'm going to make a translation between scores and something I understand better. So maybe one example of that, one particular method, might be what Draft Guidance 4 calls the illustrative item, or used to be called content-based interpretation. Let's pick an item from the measure, a multi-item measure that has multiple items thought to reflect the same underlying thing, and let's sort of get one that's really tangible, like being able to walk up the stairs unassisted. And let's see how the predicted probabilities of giving each of those responses distribute themselves here. So I can just get a better idea. So instead of a 35, I can say, well, this person's pretty able to walk up the stairs. Likely they're going to be able to walk up the stairs unassisted. That's one little clue to me about how they might look, right? So it's mapping scores to something I understand better. Other methods take a different strategy, and they are mapping differences between COA scores to some other differences that I understand better. So here one example might be, and now look, the score here is a difference score, so it's centered at 0, right? I might try to map that now to, I'm getting some funky, there we go. I might try to map that to the patient's verbal characterization of their change that they experienced, right? And in everyday language, our tech colleagues, there's a bunch of windows open in front of my slide that you guys can't see that, but okay, thank you so much. Maybe we do the formatting later. Okay, now we can see. What I'm trying to do there is to take some language that's a little friendlier to me, because we might tell one another, I'm a little better, or I'm a lot better. I can relate to that better. So I'm going to try to map that to some score differences, right? So we've got these two different strategies. These are just general strategies that could describe old approaches, current approaches, or yet to be developed approaches, but it just sort of helps us locate the general approach being taken, right? And also helps us think about the situations where some methods might be better adapted than others. And then we'll be talking about that a little bit today. Regardless of which approach you use, the criteria for the quality and rigor of any of the specific methods in a context of use would include how well the assumptions are met in that context of use, how well it reflects the uncertainty in its results, because we don't always know, right? And one of the things we want to do scientifically is to characterize our uncertainty, and how easily decision makers can use the results to better understand how a patient's life might differ with and without the treatment under consideration. Right? Furthermore, if we were translating a text and I said, well, I can give you one translator, or I could give you 3 different translators, I want 3 different translators, because different translators might be better at describing different aspects of this, right? And so multiple translators, multiple methods may provide a richer context for interpreting the treatment effect. The result of applying any method or methods will inform our belief that the treatment's doing something meaningful to patients, right? But that belief is also informed by other things, such as the results from other endpoints, safety signals, context of use, etc. Right? And that's really important. We need to manage our expectations. There's no analysis we'll do of one endpoint that's going to kick out the conclusion that this treatment has a meaningful benefit for patients. That's not the way we come to those conclusions, right? So, big summary here. Interpreting treatment effects on COA-based endpoints often requires methods for translation of scores into something more relatable. Over time, the field had narrowed its choice of COA-based endpoint and the method of translation, but these approaches are not always the best fit for our situation. Draft Guidance 4 invites greater flexibility in selecting, developing, and applying methods to support COA-based endpoint interpretation. And those different methods could be categorized by the approach they take to translation. Either I'm going to translate scores into something that's more relatable, meaningful score regions, or I'm going to translate score differences into something more relatable, meaningful score differences. Right? And the results from these methods are just one part of the evidence used to evaluate the meaningfulness of a treatment benefit. Okay, that's it. Oh, and there we go. We're going to focus the rest of the session on meaningful score regions. Okay.
All right, thank you, Kevin, for that wonderful presentation. In our preparation for this panel, in our discussions, Andrew proposed the idea that there was nuance on this subject of meaningful score regions. And that became an acronym. That was what he proposed. And that's kind of what's going to be guiding our discussion today. So each letter is going to be a discussion point. There's going to be one of my panelists who's going to kick off that discussion. There may be a couple slides to reference as well. And then that'll lead into additional questions and discussions. So moving forward, we'll start with not always required. And Kim, if you wouldn't mind getting us started.
Sure, thanks. So yeah, it's a bit of a negative point to start on, isn't it? But before I go into why it's not always required, let's just maybe for those that haven't made it to the back pages of the PFDD4, this is the meaningful score regions that we're talking about. So just to build on what Kevin was explaining to us, it's a fairly simple idea in terms of we're looking at the distribution of our COA score in relation to like a PGIS, maybe a severity measure, and we're kind of mapping those COA scores into these regions that represent severe, moderate, mild, or no severity. So it's a really neat idea. I'm a statistician. I was really excited to see this because we're kind of getting back to looking at our treatment effects, looking at our mean differences between the groups. We've spent a long time, as Kevin was saying, narrowing the field a little bit, focusing on dichotomizing our lovely continuous rich data, and that's really hard for a statistician to see. So I was excited to find this in the back pages of PFDD4, and it's a really neat solution. So the idea is you get this figure on the left. When you've done your box and whisker plots, you don't have any overlap. You can easily define those regions. Once they're defined, the graph on the right-hand side, you can then superimpose your treatment effects with a mean, a confidence interval, and you can have a look to see what the difference is between those and whether they move patients on average from one category to another. So really neat idea. Why might they not always be required? If you want to move on to my next slide. So I'm going to show you 3 meaningful score region plots from the same trial. So like everybody else, I'm sure, I saw this in PFDD4, immediately put it into all of the studies I was analysing, really excited about it. And then this happened. So we thought really carefully about our anchors, I would like to say that. Some of them didn't correlate very well, as you can see. But we did think about what the context was, what the instrument was, and which anchors might really closely match the same concept. It's a hard situation to do this in fairness. So this is a performance-based test, and it's the patient's own rating of severity. As you can see, we had a really poor correlation. It was 0.1. It didn't really work well as an anchor at all. So as a result, our box and whisker plot looks nothing like the beautiful one in PFDD4, completely overlapping. Because of the trial entry criteria, just to note that a lot of our patients were in the kind of none and mild. So there's not many people in the severe category, but you can see there's just a huge variation in the scores, whichever category patients were placing themselves in. So scrap that one, and we moved on to our next anchor, which did perform better. So this is now an interview-based assessment, and the interviewer also filled in a separate global rating of severity. So we had a good correlation for this. So when we're looking at our anchor correlations, as long as it's 0.3, we're kind of happy. If it's 0.5, that's great. That's much better for us. So it's sufficiently correlated as an anchor. But when you look at our box and whisker plot, we'd still have some problems in defining regions based on those. There's still a lot of overlap, despite that good correlation. You could see if we tried to put a boundary between the none and the mild, we'd have half the distribution from one overlapping in, and it really wouldn't be a very good cut-off. You could potentially put those categories together and say we can't distinguish between the none and the mild or the moderate and the severe, but we could have two bands maybe. But then if you remember, what we're trying to get back to is what on average are our treatments doing, and are they moving us between those bands? And then you've got this huge hurdle to cross with your trial. So our next example, this looks more like it should do, doesn't it? This is an interviewer-based assessment again, but within that interviewer-based assessment, they had their own little global severity scale within the same instrument. So we basically asked them the same question twice. It was really highly correlated at 0.82, as you might expect, a little bit circular, you could probably say, because it's from the same instrument. It's probably pretty unrealistic for most of our anchors to look like this. I certainly don't think when you ask patients, I don't think we see correlations up at 0.82. I think they're much more normally around 0.3 to 0.5, 0.6 if we're lucky. So I think this is highly unusual to do this well with your anchor. We could define regions using this. They're not equal regions, and the guidance states that ideally they would be equal regions, and if not, then you need to consider it further. I don't know what we're going to consider further, but yeah, you could define regions for this one. So that's a nice example. Do you want me to pause there? Yeah.
Yeah, if you wouldn't mind. So I love these examples, and I love that they're all from the same work, because we're getting a little bit of an idea of the challenge of it. But I'm kind of curious, if Kevin and Andrew, if y'all have reactions to this. Andrew.
Yeah. So I mean, I guess it's not surprising for me to see, you know, these examples in the range. There is overlap. There probably always will be overlap, and I think, you know, while we can estimate the optimal threshold and, you know, draw a picture of where the boundaries are, I think ultimately we need to live with that overlap and recognise and be able to communicate that there are kind of grey areas where, you know, actually for a given score, I can't see the numbers on the bottom of the plot, but for a given score you could say, well, actually it's really 50-50 whether this is kind of none or mild. You know, we shouldn't kind of pretend that these regions are totally fixed, and as soon as you get one extra point on the score, your whole experience massively changes. So yeah, it reflects my experience, I think, these examples.
Yeah, I think that, you know, if we go back to the basic logic here, the reason why these anchors were used was because we thought that maybe that language of, say, mild, moderate, severe, or whatever the terms are going to be, that's more accessible language to us. And so if someone told us that they were feeling moderate, their level of activity is moderate or low, we would understand that a little bit better than if they said a 34. And so when we see low correlations, you know, what we're seeing is that people are not consistently using these terms. They don't use them the same way, and we all know that, right? We all know someone who would say they're in severe pain, and you know they're a little bit of a drama person. So there's variability in how people use these terms, and that's when the more variability there is, the lower the correlations and the less useful those terms are for us as mapping devices. It's interesting, by the way, to contrast this with a bookmarking study where you explicitly get a group of people to come in and reach consensus on the meaning of those same ordinal terms in relation to some vignettes, right? There the correlation is 1.0 because you all agreed on what it is. But when we don't have that and we're just going with the way people are naturally characterizing their lives using these ordinal terms you gave them, we find out sometimes it's not used consistently enough for us to use this as a tool to get a better understanding.
Great, thank you both. moving right along and keeping going. So Kim, why don't you wrap us up on that?
Yeah, so we started off with why it might not always be required, and I think there's some thinking to do around where they're going to work and where they're not going to work. So I held off telling you what the disease context was in this. We were measuring cognition using various performance-based tests, interviewer-based tests, and we were asking patients to rate their own cognition when their cognition was possibly declining. So that's a difficult thing to do, isn't it? So I think maybe then they're not always appropriate, and I think in some disease areas, or depending on what the expectations of treatment are, maybe they're not a useful tool. I think, for this, we were looking to slow the decline rather than improve the condition. And if we'd have used that last graph and put the treatment means on that graph, we probably wouldn't have shifted anyone from one category to another. But in terms of whether the trial was successful or not, it might well have been hugely successful if we maintained people where they were. They weren't progressing. So I think, as we saw in that as well, even when we have really good anchors, we may struggle to define those regions. And then, as Andrew was saying, it feels like quite a high hurdle, doesn't it, to say, well, one treatment should be in one area and the other treatment in another. So do they make more sense where we expect a treatment to improve a symptom or a condition? You could think of some examples, say, with a knee injury, you're having surgery for the knee injury, you would fully expect that a lack of function before surgery should get better after surgery. So something like that, you're returning someone to something that resembles normal function. You could see that you are looking for, if you're comparing two different treatments, you're looking for them to move you along those regions. Something like asthma, are you controlling the asthma versus uncontrolled? Eczema, are you returning skin to normal, or is it still in a flare? So those kind of settings, maybe this works much better and is a useful tool. I mentioned the trial inclusion criteria, so ours was kind of limited to patients with none or mild cognition impairment. But does that limit the application of the meaningful score regions if we have these narrower patient populations? So we may well be in the situation where we're defining two bands, and they're quite large bands going across the COA scale. So how useful is that going to be? And also, as a final point on this, just in terms of, can we really expect our anchors, as we traditionally have them at the moment, to be strong enough to define the regions? As Andrew was saying, we know there's uncertainty about where those boundaries are. It's never going to be perfect. But actually, you need a really strongly correlated anchor to get anywhere near certain as to where those regions should be. So typically, we don't have those strong anchors in our studies.
Thank you. I mean, this brings up a couple of points. One is, does it make sense to pull these numbers from clinical trial populations? Is that the correct population to look at when we're developing these numbers? Andrew, you look like you're ready to answer.
Yeah, I would say yes, because those are, you've got the right context of use. You've got exactly the right types of patients for whom you're trying to make a claim around the meaningfulness of any treatment effect. So yeah, absolutely.
And I think another question I have is, I think you gave us a little bit of information about how we're deciding when this is and isn't appropriate. Like, there's a lot of context within your context of use that can help drive that decision. But is there anything else we should be taking into account when we're kind of deciding whether or not this is appropriate for that given context of use?
I think ideally, early in the program, you would have engaged with patients to find out how they would construe benefit. And if they're construing benefit as, I need to get to this place, a treatment would need to get me to this place, that would be more consistent with an interpretation that's based on trying to map the endpoint to a particular health state, right? As opposed to how much of a change would be a meaningful thing. So I think trying to get some patient input about how they would construe benefit could help inform the selection.
Great, thank you, Kevin. And I think, yeah, that really fits in with Kim's examples of kind of knee surgery or asthma, where that might be how they think about their current condition and what treatment might go. So moving on to understanding and explaining your rationale, right back to Kim.
Yeah, so that moves us nicely into the rationale for the inclusion or the not inclusion of your meaningful score regions in particular studies. So Kevin's covered the first point here. So how are patients talking about their disease treatment? What's the expectations of this treatment, of this study? And that's a really good starting point in terms of, are these going to be useful for us? Because even if we can define regions, if we're looking to maintain people in a stable state, then we're not going to see that shift across the categories. I think up front you could have a rationale for not including the meaningful score regions. We discussed some of the reasons on the previous slide as to why they might not work well in a certain situation. So you can make decisions based on your expectation of the disease course, the treatment impact on the disease course. But I think also it's a multi-stage approach. So I think you might, with the best intentions, include meaningful score regions and then find that they're not going to work well in your study. I don't think you know all of these things up front. So I think a worthwhile approach is to be thinking about upfront whether they should be in there, but then have a look at your anchors. How are your anchors behaving? What do these box plots look like? Can you reasonably define those regions? And at that point, you might have a no-go. This isn't going to help us with our interpretation after all. And then I think a stage further, as I showed, you might have a really good, strong anchor correlation, and then your box plot can still look really horrible or give you a massive hurdle that you know isn't reasonable. So I think these are all points at which you could have rationale for not including the meaningful score region at that point. And I think we're going to touch on anchors a little bit more as we move through the nuances. But just in terms of that choice of anchor, what is the right anchor for defining the meaningful score regions? Like, we've lent very much towards the patient ratings recently. Is that the right thing for this setting? Should we be looking at accepted cutoffs for some diseases that are already used in clinical practice? Could that be an acceptable use of an anchor for this purpose? And importantly, the qualitative data to support the choice of that anchor, I think, is going to be super important, and the meaning of those categories and the meaning of having one treatment moving you between those categories.
Thank you. Now, we've heard the PFDD guidance documents mentioned quite a bit. I mean, they're kind of the elephant in the room for the last few years. And Kevin, I'm kind of curious, do you have anything about, could you say anything about how this might fit into the evidence-based rationale in Guidance 3?
Sure. I think actually Dr. Lily Gerard made this point earlier today, in that, you know, one theme I think is just trying to think about what makes sense and then explain your thinking. Right? What is the rationale for the decisions being made? What other courses were considered, and what's the reasoning behind the choices that were made? That's the whole point of the rationale-based approach from Guidance 3, and it's the same thing for trying to explain the justification for choosing the endpoint that's constructed. And similarly, you need a rationale for how you're interpreting this result in the context of other things. And so I think that's a theme that kind of cuts across, is that we need to think carefully and then present the thinking in a coherent story that's well supported.
I love that reference to telling a story. It's when I think of validity theory, that's kind of how it plays out in my mind. We've already talked a little bit about anchors, but we'll move on, and next is the fact that anchor selection matters. So, Andrew, could you tell us a little bit?
Yeah, thanks. Seems really loud, but yeah, so I want to focus first on regions defined by a PGIS, or a patient global impression of severity. And we need to be careful with this, because if you advance the slide, And if we look carefully at what is in the draft guidance, I'll read it so I don't mess it up. But so if the treatment effect is larger than the width of the widest scored region, this suggests that the treatment effect reflects a meaningful difference. By treatment effect, if we go to the next part of the slide, this is in terms of a continuous mean difference between two treatments, where really the length of the line between the kind of treatment and placebo boxes is the treatment effect that we talk about here. And if we take this wording, literally, then we are comparing the length of this line between the two, and we're comparing it to the width of the widest score region. And on the next slide, I kind of question that approach, really. So, sorry to give you a formula this late in the day, but I think it's quite a kind of useful one, because, we're asked to consider the maximum meaningful score region width. And you can find out that the minimum that this will ever be is the scale range of the COA divided by the number of anchor categories. So for example, if we're using a 4-category PGIS, sometimes we use a 5-category, but a 4-category PGIS that's in the guidance, if you've got a 0 to 10 scale, like a pain or itch numerical rating scale, you'll need to see a between-arm mean difference of at least 2.5 points. For something like the EORTC, which has a 0 to 100 scale, you need to see a treatment effect of 25 points. For anyone who works in these areas, these are very, very big treatment effects. I looked at kind of meta-analyses for pain and itch outcomes, including the FDA approved. None of them for pain met the 2.5. There was one study for itch that I could see. For the EORTC questionnaire in cancer, there are published guidelines around a meaningful between-group difference between arms. They're kind of categorized as small, medium, and large. For all but one domain, even the large estimates are below 25. So I would say, really, to exercise caution, if we're using a 4-category PGIS, to not be, it's a unrealistically high hurdle, is what I would say, if we are comparing the treatment effect to the maximum width, as is suggested in the guidance. So on the next slide, I would say, well, it's two things, really. So first, anchors are not the only option to estimate meaningful score regions, but also we need to have a think about, okay, well, if we don't want to be or shouldn't be comparing the magnitude of the treatment effect to the width of the regions, what should we be doing with the regions, and how should we interpret a treatment against them? And Kevin talks about that a bit later, but I will just say here that, yeah, anchors are not the only option. We talked about qualitative, we talked about bookmarking and stuff like that, but I think really with those approaches, you want to get it so we're having more granular regions. Like in an ideal world, I would be able to tell you what every single unit score meant. So we want to get kind of as close to that as possible. So I would say, yeah, we need to kind of rethink the anchors a little bit and start thinking about being a bit more granular than what we're used to. And yeah.
Thank you. Yeah, so you mentioned kind of looking at this qualitatively, and I think, Kevin, you're going to touch on that a little bit later, if I'm remembering correctly. But, well, not really, I guess. But but also bookmarking. And I'm just curious about these other options besides using side of PGIS, PGIC, if anybody on the panel has kind of used these yet, and if anybody has any thoughts. Oops, that was the wrong button.
So it wasn't called bookmarking in my day, but when I did my PhD many moons ago, we were talking to clinicians and patients about group changes, and that's the paper Andrew referred to for the URTC. So we were doing a similar thing in terms of saying, on average, the patients in this group are around 50 points. If you were around 50 points on this scale, these are what your responses might look like across the items. So we were doing a similar kind of thing a long time ago, just trying to kind of benchmark it with where you would be on those response scales. So I have some experience of doing that. What I would say is that was a PhD. I did it part-time. It took about 10 years. No one's going to do that when they're developing an instrument or looking at their clinical trial program. So it's a hard one to think about how do we translate into something we can do in a reasonable amount of time for a reasonable cost. I would say it did work well. I think when we debriefed with patients, it was something they understood. And they felt they were able to compare the difference between groups, which is quite a different thing to ask patients to do. So I think it's possible, but I think we're not quite there yet in terms of how would we do that in a timely and a reasonable fashion.
Thank you. So, I mean, it sounds like we have other methods, and some of them, I think, have come a long way and are more streamlined these days, and I think some of them are still being developed. And it's important that we keep our eyes open and keep reading about that as we move forward. So moving on to the non-transferability. Andrew.
Yeah, and I'm going to talk quite quickly about this one, really. So I think it's just this idea that, if we have estimated thresholds between regions or severity cutoffs in another population, can we transfer that to our context of use? And I think it's, the answer to that is the same with anything else, and it's there's levels to it. Like, I think if it's a completely different disease, then no, probably not. Whereas, of course, if it is, the same indication, there is just a slight difference in the eligibility criteria, then it may be more reasonable to do so. So yeah, it's a quick one for this point, really, but it's just just touching on that subject of the extent to which we can kind of transport the thresholds from one area to another.
Thank you. So Kim earlier gave the examples of knee replacement or eczema, and it's the idea of bringing scores up to a kind of certain level. Maybe getting you up to high function or total clearance. And when you see those examples of treatment bringing you closer to a certain level, is the expectation kind of to bring them to a level of like a population level? And might that be something that's kind of somewhat common across some diseases?
I think that's a really difficult one, isn't it? I think it would be really nice if we could have, say, compare how we're improving fatigue across different disease areas, and for that benchmarking of fatigue as normal, say, is the same across those disease areas. But I think the more we look, the more we find it's more and more nuanced, isn't it? It depends where you're starting from. It depends what disease you're living with as to what's an acceptable level of fatigue for you. So I think ideally it would be nice to be able to say we could define regions on a concept and then apply them across different disease areas and make use of things like normal reference populations. But I think the reality within the trial context of use is that you're in a narrower band and you have to consider where people are starting from. So yeah, I think that's a really tough question.
No, thank you. And I think Michelle's going to probably smack me later because we've got the t-shirts that say it's the context of use that's important. And so we got to keep that in mind. All right. Moving on. Kevin, I think you referenced this as the illustrative example item earlier. And so if you wouldn't mind saying a few things about this.
Yeah, so back in the 90s, there were, as I mentioned, lots of different ways that people were proposing to create some context for the scores, and one of those was called the content-based interpretation. And John Ware and San Keller had a book chapter that discussed this, and I know Joe Capillari and colleagues have summarized it too as content-based interpretation. And the idea is that you actually just look at the content of the items and try to figure out whether that's an effective communication about where people are. And back when John Ware and San Keller had that chapter, they said, you could do this with a single item, and a single item might have response options that are very clear. That would constitute an effective communication if we were talking to someone and they responded with that level. We don't need any additional work, and I think it's noteworthy that in Draft Guidance 4, that whole section of interpretation starts with a consideration of when is extra work required to support the interpretation, and noting that sometimes, if you've got a single item and there's been cognitive interview work done that's good and that patients regard the response options as distinct and meaningful, it's pretty clear. If you have a multi-item measure, another option becomes available. And in Draft Guidance 4, it's called the illustrative item method, but it's just as content-based. And the idea is that if I have a measure, a multi-item measure, and it's the sort of measure where all of the items are thought to reflect the thing that I'm trying to assess. So they're all reflections of physical function, they're all reflections of fatigue. Then what I could do is to select one or more of those items that are especially tangible, right? They're especially easy to get a sense of what's going on, like whether you can walk up the stairs not at all, with assistance, without assistance, right? That's really concrete. And you can look at the distribution of the expected response probabilities by score level, right? As a clue to what sorts of things someone might be able to do, for example, at different score levels. And when we put together vignettes with these types of reflective measurement model measures, we're essentially doing that, right? We're getting the predicted item responses and then turning it into a story, right? And so that's one method that's available when the measure is a reflective measure. And the nice thing is the items are reflecting the thing of interest. The items should have gone through cognitive interviewing so that we've got some reasonable expectation people interpret them consistently. And I could use more than one if I wanted to, right? And it could be the case that I've got multiple items and I may do something like, well, let's look at the score levels at which you've got a greater than 80% chance of being able to do this activity, right? And I could plot that for all the different items along that score level, and this is the granularity that Andrew's talking about. Now I'm mapping those scores to the things that people might be able to do at the different score levels, right? And it just helps me to see then, if I've got two people who have the same score at baseline, where they're going to end up on treatment versus control on that scale, and I can look to see, well, these are the sorts of things you'd be able to do that you couldn't do if you were in control, right?
Yeah, thank you, Kevin. This kind of makes me think, in your example, Kim, the one thing that worked best was the actual item from the measure that you were using. And so is that the idea that maybe that one item did a good job of, in education talk, we talk about discrimination, how well it differentiates between people with different levels of the construct. And so do you think that's the case, where just that item had really good discrimination of people across the construct?
I feel like it essentially asked the same question as the question that just filled in. So we were talking about this with EURTC in terms of how do you put that really concept-specific anchor into your study without asking the same item that you're trying to ask anyway. So yeah, I think that's basically it. It was very circular, and that's why we had a correlation of 0.8. And we talk about, the higher the correlation, the better. But as you approach one, you're asking the same question, and it's not really a separate question, is it? If that made sense.
So the danger here is that we may have really pretty, just great-looking regions, but it may be because we asked the same question twice. All right. On that note, we'll move on. And Andrew, if you wouldn't mind talking a little bit about evaluating this uncertainty. I think Kevin, and I think both you and Kevin have referenced that about the need to keep that in mind this whole time.
Yeah. So I think the uncertainty, we can stay on the, actually no, here's fine. So the uncertainty comes from two sources really. So there's uncertainty in the estimated treatment effect or the average outcome on each treatment. And then there's also uncertainty in terms of, where does the boundary between regions lie? And there's uncertainty in the sense of, for a given score, you're not 100% going to be moderate. There's always a bit of uncertainty, like, you are most likely to be moderate, but you could also be mild, you could also be severe. There will be some patients with that given score responding in, all three of the different categories. So, so I've brought up here on this slide an application essentially, which is similar to what's in the FDA guidance, but with a tweak really. So the top half of the plot is really the same, where we are, plotting the mean and confidence interval, adjusted score estimated from a trial for two groups. We've got treatment and placebo here. But on the bottom it differs because rather than, these fixed blocks of regions which are kind of absolute, we reflect the uncertainty and actually that, you're never 100% certain that one region applies. And, with an app like this, you can, adjust where you think the regions might be, the boundaries between them, and kind of think about this uncertainty and how it affects your conclusions. And then if you go to the next animation, really the goal here is to, you want to be able to know for each possible score, what does it mean? We have the uncertainty here because we can click at different parts of the treatment effect, where that lies, not just the mean, but, around it, either side. And then we can also get the uncertainty in the regions by looking at the, here we see that, you're kind of 60% sure that they're mild at this score, but around kind of 30, 40% that they could be moderate. So it's about, I think, moving away from just comparing the between arm difference or your treatment effect to the width of a region, but still keeping the regions, but instead shifting to this idea of, okay, well, what are things like for people on treatment? What are things like for people on placebo? And understanding each kind of score and what that means, rather than kind of trying to map the difference onto the regions. So yeah, that was it for the last letter. So I'll pass back to Fraser.
Yeah, thank you, Andrew. I mean, that's an interesting way of looking at it, and I think a really promising way of looking at it in terms of the likelihood of being in a certain region and thinking about that in terms of your treatment and your control or your individuals of where they land. So thank you. All right, we are coming up on the end, so I'm going to leave it with kind of final thoughts for everybody so that we can get into questions. And you're welcome to pass, but if you have any final thoughts that you'd like to add, we can go with that.
I'll pass.
Passing? Okay. Good time for questions? All right, let's just move on to questions. And it's questions, not badgering the panelists. Okay. You have great questions. I'm sorry I gave you a hard time.
I think you guys will appreciate this question, all right? You guys are methodologists and are experts in the development of how do we interpret our scores that we see in these score changes. However, not everyone who publishes or makes interpretations of trial results and these methods that we're using know what they're talking about. But the publications happen and they rise to the top. And so we get criticisms that, well, that is not meaningful change. It did not help a patient, right? I don't know why a regulatory decision was made, right? We're seeing this across the board. How do we, as a field, particularly with methodologies, as we're moving from something that is kind of hard to understand and nuance unless you're in it every day with meaningful score ranges and meaningful score differences, right? That's not a norm for people, and you're struggling with basic meaningful within patient change, right, concepts. How do we as a field, and particularly as the methodologists who are embedded in this, really think about how we need to think about as we're communicating this, so we're not having to go and explain decisions we're making or why actually we are measuring the right things, because those aren't the papers that are rising to the top. It's the other papers that are rising to the top that say we didn't hit minimal clinical important difference. I would ban that word if I could, okay? I do yell at people when they say it, because we are actually missing, yeah, Fraser knows because he's heard me do it, because we're actually still even there and probably missing something important. So I turn to you as methodologists where they're now coming and kind of criticizing your work without understanding what they're actually doing when they make these comments. How do we help the field with, communicating actually, this was the right methods that we used, and this actually does represent meaningful change to patients?
So honestly, I think Kim's example is a great example of just being kind of out there with what it's looking like, right? It may not work great, and you can publish that final example by itself and say it's a wonderful score regions, but that lacks for a lot of information. But I mean, so turning that question to my panelists, I think, is kind of, how are we helping to give confidence to, Michelle, help me out here. How are we phrasing this? How can we rephrase your question? How are we giving confidence to the methods that we're using? How
do we as a field, because we have some great other folks here in the audience, how do we, as we are wanting to explore, as we're going to explore other ways to examine meaningful within patient change and what is clinically meaningful to patients with additional methodologies that are good and sound in science, right? How do we as a field help make sure that we are able to communicate that and have folks who don't see this every day, your HTA, right? Your book that's going for formulary to an insurance company in the US, right? Explain when there's caution, because the field is still a little weird if this is really a drug that's doing what it's supposed to do and a regulatory decision has been made. To say, here's why. Here is part of that evidence that was used to support. How do we as a field think about telling this story, going back to the story piece, of why this is important, and we are getting at what's clinically important, what is important to patients. I'll take out the clinical, I'll take out meaningful, I'll take out minimal. But what is important to patients, and this actually does reflect that.
So kind of, I think Kevin referred to it as the meaningful treatment effect of how do we show that. So I guess the question is, how do we drive the field forward? So we've done our session on nuance. We've asked people to think more carefully about this. We've given them some points to think about. I guess your question is, what are the next steps so that it spreads beyond the people in this room?
Yeah, I mean, I think, making the regions more granular and trying to think in that way and push that way, I think, I would say that the regions seem more intuitive to look at and communicate in terms of, this is a score, this is what it means to patients. So I think, definitely I welcome the regions existing, and I think, just a bit of fine-tuning on, how do we kind of map the treatment effect onto that is the kind of way to go. So I think just further further examples, sharing of case studies, discussions, and just kind of working together and sharing knowledge really on the regions to help advance.
I think building on that, what came to my mind and part what my PhD was built on was this kind of familiarity with scales. I think it's really hard with a brand new scale that nobody's used, nobody knows what the scores do, compared with some of the more legacy instruments, if you like, where people have seen lots of patients through the door that have got particular scores. They've got that kind of context as well. So I think that sort of familiarity with scales, which comes from publishing data from those scales, lots of data from those scales, the reference populations, the meaningful changes when they're developing the instrument. I think whatever we can do to gain that familiarity more quickly, then it helps you be able to interpret it, doesn't it? It's like any of the clinical measures. You don't know the meaning of reducing your blood pressure until you've seen a lot of patients with blood pressure that's been reduced and they have a better quality of life and better outcomes. So I think that's what we struggle with in the field, that we have so many scales and they're all different, and we don't have that kind of familiarity until we've used them for a long time.
Anything to add, Kevin? Or you...
No.
Okay. Yeah, I mean, I'll do the CPATH, what I'm supposed to do, which is, yeah, I totally agree with the sharing data. I mean, that's how we drive things forward. So.
Ashley Slagle, Aspen Consulting. I want to thank the panel for the discussion, and I can appreciate that MSRs are theoretically interesting. It's a new method. But forgive me, my brain always takes me to painfully practical questions. And we heard a lot of cautions about the MSRs. They don't always work. Even if you have good correlations, they may not work well. When they worked, it was circular reasoning. We won't know in advance if they're going to work, and we won't know till after the fact if they've worked or not. So I guess my question is, when we have methods that we have refined over the years, anchor analyses, exit interviews, the vignettes and bookmarking, why? Why? As I'm thinking about advising sponsors on setting up research to help understand meaningful changes to patients, this is a new kind of complicated method to them. All of the other methods have been socialized internally. The different cross-functional teams understand them. This they don't understand. And then it may not even work, and from the examples, it looks like it doesn't work more often than we would like. So I guess, can you give us a rationale for why we would move forward with this?
Yeah.
Kind of?
Yeah. So I think just one example is studies where you can't collect a baseline if people are really ill. So I'm thinking things like traumatic brain injury or like an acute stroke. Then essentially these events happen to people, rushed into hospital, randomized, get a treatment, and there's no time, and the patients are unable to complete. So you can't think about meaningful change. And then meaningful score regions are good because it then offers a way to talk about the meaningfulness of treatment effects within scenarios such as that, where you could then say, okay, well, 90 days post-stroke, let's think about, where do we want people to be? What kind of things should they be able to do? Would we like them to be able to do? And then use it that way. So I think there are some scenarios where, I don't think you really can measure change from a PRO.
I think to add to that, I think it should be seen, it's a tool amongst the suite of tools that we've already got. So I say in recent years, we've moved away from looking at these kind of between group differences, but we used to use cross-sectional anchors, clinical anchors, patient anchors, and we start to get an idea of what might a meaningful difference between groups be. And I think this is just another tool in that toolbox, and when it does work, I think it's a nice, neat solution. and it can sit alongside those other methods, because I think we're trying to estimate something that's not exact. So the more information we've got to kind of triangulate, narrow it down to a, I don't want to say region, to an area, it helps us. So yeah, I think it should just be viewed as an extra tool in the toolbox.
Could I have time for a quick friendly amendment? I would not characterize it as a method or a tool. It's a category label for a whole families of methods. So there's meaningful score differences, meaningful score regions. Whether they work well or not is going to depend upon the particular method that's used. And if you put a crummy anchor in, doesn't matter which of those two approaches, and we all see plenty of really bad meaningful score difference approaches using bad anchors that now we don't know how to interpret anything on. So I think we have to remember this is not a new method. These are just two labels for the rationales behind multiple methods, some of which are very old.
So I've just noticed that it's counting up now instead of down, so we must be out of time. No, this is my fault. It's my job to keep us in order. So I want to thank my panelists for joining me up here. I want to thank the audience for their attention.